Hi guys, my name is Avery. I'm basically a visiting student here at UNE. I'm already graduated from college somewhere in good old Kentucky. I just took the MCAT, waiting on the results from that. So no, nothing to tell yet, but we'll see. So I chose to do cases 1 and 3 for the case connections of Unit 2. So I'll go ahead and dive into case 1. Um, so basically, case 1, as you guys know, is asking us pretty much about pyruvate kinase M2, why M2 is preferred over type M1. Um, this was a really challenging question for me. It took a lot of critical thinking, but I came up with a couple different things. So we'll get started. So with the first one, how is pyruvate kinase regulated in a normal cell? So basically, just to start off, pyruvate kinase is an enzyme that catalyzes the last step of glycolysis. So basically, the conversion of phosphoenolpyruvate and ADP to pyruvate and ATP. It basically just transfers the phosphate group from phosphoenolpyruvate to ADP. So to get into it, pyruvate kinase is regulated pretty much by intracellular signaling and molecules involved in metabolism. So pyruvate kinase is activated by glucose and insulin, fructose 1,6 bisphosphate, AMP, and NAD plus. And this is when it's in the well-fed state that's activated by those things. Pyruvate kinase is inactivated in the fasting state by low glucose, glucagon, ATP, NADH, and alanine. So ATP and pyruvate are basically negative allosteric inhibitors. I found a little bit of information on that online. I thought I'd just add that in there. Alanine is also a negative allosteric inhibitor, or I'm sorry, a negative modulator. Phosphoenolpyruvate can also affect the activity of pyruvate kinase. It can basically enhance the activity of the reaction, and we kind of just spoke about that. That's one of the main components of that last reaction in glycolysis. So on to the next part of that question. This logic seems counterintuitive to glycolytic tissue. What is the rationale? So this is what I had the most trouble with. This was very hard, but after I sat down for I'll be honest, a couple hours, got up, walked away, thought about it. I came up with a couple things. I'm not sure if it's right, but I don't think the point of this is to be fully correct. I think it's just to really critical think. So here's what I got. Basically, like it says, it's counterintuitive. So you would think that PKM2 would be in a high activity state and would be very effective at promoting glycolysis because cancer cells are rapidly proliferating and most likely would need more energy to keep proliferating. But clearly this isn't the case since PKM2 is usually low activity and low glycolytic activity pretty much. So just to dive in a little bit, MYC is a nuclear transcription factor. It binds DNA and transcribes new proteins when cells are integrated and signaled to divide, and stem cells are constantly being signaled to divide. Cancer cells require all the same growth factors that normal cells require. The cancer is basically infecting the stem cells via MYC, the transcription factor that we just talked about, in order to rapidly proliferate and increase those number of cells. So the cancer preferentially infects stem cells to rapidly proliferate via the cell cycle. So here's one theory that I kind of came up with after a little bit of research and talking it out with myself. So if cancer cells have low activity PKM2, this means that there has to be low levels of AMP, because we just said before that pyruvate kinase is activated by AMP. So if it's low activity PKM2, then there has to be a low level of AMP, meaning that there has to be high levels of ATP which also would mean that there's going to be low glycolytic activity because there are already high levels of ATP. So what's the need to make more through glycolysis if there's already high levels? So that was kind of one of the theories I came up with. Well, not really me. I mean, I did some research and then kind of, worked through it. That's one thing. Another possible explanation for this rationale could be that PKM2 can be negatively regulated in response to growth factor signaling. So if this were the case, it would make sense that PKM2 had low activity because theoretically, if cancer cells are constantly, and not only constantly, but also rapidly dividing, there should be more growth factor present. At least that's kind of how I went through it in my head. Maybe it's too simple, too easy, but that's kind of the way I thought of it in my head. So those are two kind of theories that I came up with. Like I said, I don't know if it's right, but again, I think the point is to kind of critically think and see where we go with it. So then the third part of this question, what other pathways may cancer be diverting substrate to? I also had a little bit of trouble with this one, but after researching a little bit, I found, according to the Warburg effect, and I'm sure a bunch of you came across those words during this research as well, even in aerobic conditions, cancer cells tend to favor metabolism through glycolysis rather than oxidative phosphorylation, which is kind of strange because according to the internet, aerobic glycolysis is much less efficient than oxidative phosphorylation in terms of energy production. But through glycolysis, metabolites are generated that are beneficial to rapidly proliferating cells, for example, cancer cells, like what we're talking about now. PKM2 inhibits the pyruvate kinase reaction and diverts the remaining glycolytic substrates into different biosynthetic and reduced NADPH generating pathways. So that's something that I found through research on the internet. And that's kind of, in my own words, what I understood to be what they were talking about. Also, the cancer cells may divert substrates to production of vascular endothelial growth factor, increased blood flow because they need increased blood flow because basically as the mass of cancer cells increase, so does the blood supply. And another one, they could also divert substrates to production of neutralization of reactive oxygen species via production of enzymes. They could do this by diverting substrates to the pentose phosphate pathway, something else I came across on the internet, and I think that that's what the article was trying to get across to us. Okay, so moving along. Case 3. So case 3 was pretty much about a boy, his blood glucose, samples were taken, persistent thirst, frequent urination, very drowsy. So for the first part of this question, what is the information likely to suggest about the underlying biochemical basis for the patient's symptoms? Pretty much, it seems like the patient is showing symptoms of diabetes mellitus type 1. The patient is in a state of hyperglycemia because he's not producing any insulin to bind that glucose and get it out of the blood. Another thing, the tissues with insulin-independent glucose transporters can uptake glucose without the help of insulin, like insulin signaling, but other tissues such as skeletal muscle, cardiac muscle, they can't take up the glucose without the insulin. So it's kind of harmful to that. So his body is most likely secreting glucagon in response to impaired glucose uptake by the tissues, if that makes sense. The release of glucagon will activate metabolic pathways such as glycogenolysis, gluconeogenesis, and fatty acid oxidation. So the next part of this question, what would you predict the patient's fasted blood glucose levels to be given his current condition? Definitely above normal range. If he has diabetes type 1, he's not producing really any insulin, little or none at all. So his BG levels will be above normal for sure. The third part, list 2 regulatory enzymes that are likely to be active in this individual's current condition. So glycogen phosphorylase is activated by glucagon, which will start breaking down glycogen stores, and then carnitine acyl transferase is activated by glucagon and will break down the fatty acids to create the ketone bodies and acetyl-CoA, which can then enter the TCA cycle. All found from searches on the internet, and my works cited is attached on the discussion board. And hopefully we all came to a somewhat similar solution. All right, thanks guys.